Dr. Gobind Khorana's early life was marked by humble beginnings and a relentless pursuit of knowledge. He was born on January 9, 1922, in the small village of Raipur, Punjab. Khorana grew up in a modest household. Despite financial challenges, he displayed a remarkable aptitude for academics from a young age. Encouraged by his family emphasis on education, Khorana excelled in his studies and eventually pursued higher education at Punjab University. His academic journey continued with a master's degree in chemistry, followed by a PhD in organic chemistry from the University of Liverpool in 1948. This solid educational foundation laid the groundwork for his illustrious career in scientific research, which would ultimately lead to groundbreaking discoveries in genetic and molecular biology.
Gobind Khorana Nobel Prize-winning work in the 1960s revolutionized molecular biology by deciphering the genetic code. Khorana synthesized short DNA molecules, oligonucleotides with specific sequence representing different codons. 3 nucleotide sequence in mRNA that encode amino acid. By analyzing the proteins produced, Khorana determined which nucleotide combination correspond to specific amino acid. His research unveiled the universal rules of genetic translation, showing how DNA is transcribed into RNA and translated into protein across all living organisms. This groundbreaking discovery earned him the Nobel Prize in Physiology or Medicine in 1968, shared with Marshall W. Nirenberg and Robert W. Hall, and laid the foundation for advances in genetic engineering, biotechnology, and medicine.
I'm going to share about application part. There are 4 main applications that are inspired by Gobind Khorana's discovery. These applications including biomedical research, molecular medicine, synthetic biology, and biotechnology and agriculture. The first one is biomedical research. Khorana's breakthrough in decoding the genetic code has provided a roadmap for understanding the translation of DNA sequences into proteins. This foundational knowledge has played a critical role in investigating the patterns of gene expression across various biological processes and diseases. Molecular medicine. Khorana's work contributed to the development of synthetic nucleotides. Synthetic biology. Synthetic biology is a new evolution of biotech innovation that enables biotechnologists to go beyond manipulation of 1 or 2 genes and apply engineering techniques to make changes in entire genetic pathways. And the last application is in biotechnology and agriculture field. Khorana's insights into the genetic code have been applied in the development of genetically modified GM crops with improved traits.
Now, I will explain about the advantages. The first advantage is advancement in disease understanding and treatment. Gobind Khorana's groundbreaking work in deciphering the genetic code significantly advanced our understanding of how genes detect biological processes. The next advancement in disease understanding and treatment is polymerase chain reaction, PCR. The principle of genetic coding elucidated by Khorana laid the foundation for the development of PCR. The second advantage is innovation in molecular medicine. Khorana's creation of synthetic nucleotides was a pioneering step in molecular biology. He developed methods to chemically synthesize oligonucleotides, which become essential tools for understanding the genetic code. Thirdly, advances in synthetic biology. Khorana's work on synthesizing oligonucleotides laid the groundwork for synthetic biology, where entire genetic pathways can be engineered. The last advantage is technological progress in biomedical research. Techniques like microarrays and RNA sequencing rely on the comprehensive understanding of the genetic code, which Khorana helped elucidate. The first disadvantage is ethical concern of gene manipulation. The ability to manipulate gene, an outcome of Khorana's pioneering work, raises significant ethical questions. The second disadvantage is safety and unintended consequences. Genetic modification can result in unintended consequences, such as off-target effect where non-target genes are altered. The third disadvantage is access and inequality. Advanced biotechnological treatment, which derive from the foundation Khorana laid, are often costly and may not be accessible to everyone. The last disadvantage is regulatory and oversight challenges. The swift advancement in synthetic biology and genetic engineering enabled by Khorana's contribution often outpaces regulatory framework.
This is one of the case study about Gobind Khorana, titled Gobind Khorana and the Rise of Molecular Biology, written by Chris Kaiser and published by MIT School of Science. The MIT School of Science is the global leader in conducting basic research at the scientific frontier. The starting of the case study is about introduction and MIT's role in molecular biology. The rise of molecular biology, about DNA's role. DNA, the molecule carrying genetic information, was discovered to be crucial for cellular processes and inheritance. Emergence of molecular biology. As scientists unravel DNA structure and function, molecular biology emerged as a discipline focusing on understanding how genetic information is stored, transmitted, and expressed in living organisms. Then, about MIT's contribution. The strategic recruitment. Second key highlight about the case study is key scientific contribution. First is Alex Rich. It's about nucleic acid hybridization. Rich discovery of nucleic acid hybridization allowed scientists to identify and study specific nucleic acid sequence, paving the way for genetic analysis and manipulation. Second one is Salvador Luria. Bacteriophage studies. Luria research on bacteriophage provide a model system for studying genetic processes like replication, recombination, and mutation, leading to fundamental discoveries in molecular genetics. Next, the third point of the case study is about how Gobind Khorana's breakthroughs. First, cracking the genetic code. The synthetic nucleotide sequence. Khorana's innovative use of synthetic chemistry to create nucleotide sequence helped decode the genetic code, and monumental achievement in molecular biology. Then, synthesizing genes. Alanine tRNA gene synthesis. Khorana's successful synthesis of the alanine transfer RNA gene demonstrates the feasibility of synthesizing functional genes, opening doors to genetic engineering and synthetic biology.
For the fourth point is technological innovation and legacy. The innovators is Marvin Caruthers, and the innovation is DNA synthesis. Automated synthesis methods. Caruthers' development of automated methods for synthesizing DNA and RNA revolutionized genetic research by making DNA manipulation more efficient and accessible. The second one is phosphoramidite method. This method simplified DNA synthesis, enabling researchers to synthesize custom DNA sequence for various applications in molecular biology and biotechnology. The last one is impact and future direction, which is advancement in molecular biology. Khorana's contribution significantly advanced our understanding of genetic process and paved the way for practical application in genetic engineering, biotechnology, and medicine. The second one is inspiration for scientists. His visionary approach and groundbreaking achievement continue to inspire scientists to push the boundaries of molecular biology, leading to new discoveries and innovation.